Hi everyone. Are you ready? Yeah. Okay. In this video, I'm going to explain DNA replication, but with a little more details on some names of the enzymes which are responsible or which are involved with the DNA replication. So what do you think when you look at this picture from these areas? Pause the video and let me think about these areas and what makes you thinking differently from this slide. That's right, you guessed correct, because I know that you'll remember the differences between eukaryote and prokaryote, and here there are multiple places for eukaryotic DNA in order to be replicated, since the DNA length is much longer than prokaryotic DNA. So each of these areas are a place for originating or initiation of a replication. So this is happening in the eukaryote because they need the replication faster and more efficient. These points are known as origins of the initiation, and will have the same or very similar base sequences. So, which means that there is a place that they will be recognized for initiation of the replication, and they have a similar basis. Okay, proteins called origin recognition complexes. So the proteins that they can recognize this area, and in order to do recognition and starting to do replication in multiple places. So these proteins are called origin recognition complexes that they can bind, and then after they bind, then DNA helicase will start to separate the DNA at these places. So let's say again, the DNA, the complex will recognize the area, let's say here, or let's say here, or let's say here. The complex will recognize here, and most of these they have a similar basis. And once the complex was recognize the area, then the helicase will attach and start to separate, and the helicase will attach and start to separate. Helicase can start to do the separation, or if this recognition is happening here, or it might be here. So the thing is, from this picture, it depends on the movement, that I suppose that the movement or the replication direction is starting from left to right. So the helicase also is trying to separate to unwind the two strands from this direction. Okay, in this topic, prokaryotic DNA replication is examined. So this means that once you see in these slides, all of them are discussing on prokaryotic DNA, not eukaryotic. And prokaryotes have a single replication fork, and they have a simpler mechanism for replication, not the same as eukaryotes. This is a helicase. The helicase that has started in this picture. The helicase has started from right to left. So it depends on the place or the direction of the separation, the direction of the replication after separation of the strands. The helicase start to move, and here, as you can see, helicase is here, has separated the two strands, and these are the original DNA or parent strand or original DNA. So the DNA helicase start to unwind the two strands, and then it separates, and then by the usage of the ATP and the presence of the polynucleotides and also the polymerases, then the replication happens. Again, here you will see that the helicase has started to unwind the DNA, but also you will see the new strand has been already started to make, and the enzyme which is responsible for making the new strand of the DNA is DNA polymerase, which creates the complementary strands. Look at this box. This is important. For HL students, you need to know the differences between DNA polymerase 3 and 1. What you can observe from here, and all the function that you already learned for the DNA polymerase was DNA polymerase 3. So DNA polymerase 3 was the one that you have already learned, and that's the one that which adds the polynucleotides in both leading and lagging strands. Okay, but you will learn the function in the process of replication of another DNA polymerase, which is called DNA polymerase 1. We will talk about it later, as it's mentioned over here. We'll talk later. Okay. So another thing which is important, and you will make that rule for yourself, is that the direction of the helicase is the same as the direction of the 5' to 3' for the continuous polynucleotide strands or for a leading strand. So if the helicase has started to unwind from here to here, from right to left, then this would be 5' to 3' continuous strand for which we call it leading. That's the leading strand, continuously will be made. And this is the rule that you make for yourself, that you don't, you never get confused about that. 5' to 3' is the same as direction of the helicase movement. So then in this strand, which is lagging strand, and it is not mentioned here, and even it did not shown the Okazaki fragments, the polymerase, DNA polymerase 3, it makes the nucleotides as DNA as Okazaki fragments like this, from 5' to 3', which is in these stations, which is at this direction. So this is a 5' to 3' for each Okazaki fragment. Okay, so this is a rule, and we follow this rule, and we never get confused about that. And as you see over here, the DNA polymerase always moves from 5' to 3'. The movement, okay, the movement is 5' to 3', and then it catalyzes the covalent phosphodiester bonds between sugars and phosphate groups. And DNA polymerase also does the proofreading, which means that consequently, mistakes are very infrequent, occurring approximately once in every billion base pairs. 1 in every billion base pairs. This is because this is very rare, and this is because the DNA polymerase also does the proofreading, and very rarely happens the error in the replication process. What I explained in the previous slide, still it is available with this slide with details that I just explained. Let's say this is the helicase. So let's go back to our rule so that you don't get confused. The helicase is moved. This time shows that helicases are moving from left to right. So the continuous strand, which is 5' to 3', would be this one, which is with the same direction, from 5' to 3', same direction with the movement of the helicase. And of course this would be the leading strand, and this would be the primer at the beginning of the leading strand. And the opposite strand would be, which is not at the same direction with the helicase movement, would be definitely the lagging strand. And lagging strand, as you know, it has more than one primer. Here is only one, but here you can see in each Okazaki fragment at the beginning there will be one primer, and again one primer. And then DNA polymerase 3, which after primer attachment starts to make to the near strand by adding the polynucleotides. And this is one Okazaki fragment. This is one Okazaki fragment, and this is one Okazaki fragment. Here is the point that you have to concentrate and focus more comparing to your previous knowledge, and that's the presence of the DNA polymerase 1. So this is what is necessary for you to remember as the function of DNA polymerase 1, which it shows here. The function is removing the RNA primase. So because this not only is an RNA, but also it is something which is, the function is not for the purpose of replication. The function is just for recognition of where to start the replication. And also it is RNA, it is not DNA, so it has to be removed. DNA polymerase 1 function is removing this RNA removed. So it removes this RNA, and then it adds the new DNTP or nucleotides after removing. So the next step is, after this DNA polymerase 1 is functioning by removing the RNA and adding the DNTPs, then you will see that it looks like this. So the nucleotides have been added to this place, but you see the connection between nucleotides has not been made yet. So there should be the bonds necessarily between these nucleotides, and that's the function of DNA ligase. The DNA ligase is attaching or making bonds between these nucleotides after DNA polymerase 1 added. Okay, and another enzyme which is here, RNA Primase. RNA Primase starts at the beginning, which means that by adding RNA to the place which is the place for recognition, or which is the place for starting the replication. So RNA primase add the primer, and then DNA polymerase 3 recognize this primer and start to add the DNA, start to add the polynucleotides. We have explained and we have seen this slide exactly before, and the important is, why do we call 5' to 3', and why do we know as the direction between 5' and 3', and what does that mean? Is because on each nucleotide has 3 places for making a nucleotide. One is nitrogenous bases, the other one is sugar, and the other one is phosphate. And in this sugar, which is a pentose sugar or ribose, and more precisely it is deoxyribose, not oxyribose, which is has been lost. It has been lost, or it lost the oxygen here. We have the carbon numbers starting from here, 1, 2, 3, 4, and the carbon number 5 is here, and then attaching to this carbon number 5 is the phosphate group. So 1, 2, 3, 4, 5. So based on these numbers, you will see that every nucleotide at one end has a carbon 5, and in the other end, which is 3, from 3, it will be attached to another phosphate, as you can see over here. So there will be always a carbon 5 at one end and carbon 3 at the other end. By naming as prime, by adding prime to the 5', 3', just for making a difference in names of the numbers, which is related to the carbons inside the nucleic acid, then there will be always a direction of 5' in one side and 3' in the other side. So that's how we mention the 5' to 3' direction. Well, I think I have already explained the function of the enzyme. So RNA primers, which will be added by RNA primase. RNA primer will be added by RNA primase, and it will be a place for attachment of the DNA polymerase 3, the place for recognition. And RNA primase consists of a short sequence, usually by 10 base pairs, and it has the RNA nucleotides. It's not DNA, it is RNA. And the function of the RNA primase, as I mentioned, the DNA ligase, and then DNA polymerase 1, and so on, which means I just explained in previous slide. And the adenine attachment, or making a complementary with thymine and guanine with cytosine. It has been explained quite a few times in the past slides. And also the explanation regarding semi-conservative, and you have seen completely their experiments regarding proving the semi-conservative. Pause the video and tell me who was the name of the scientist about the semi-conservative experiment. Great. I don't tell you, you should have guessed by yourself. And again, another slide explaining the same thing with different picture. Something it is new, and if you look at this replication fork, the new thing is you can also make again the same rule, movement of the helicase at this direction. So then the continuous The continuous one is the one that is moving with the helicase. In this case, would be this strand as a leading strand, 5' to 3', and the lagging strand, which makes the Okazaki fragments over here. The thing that here you will see as kind of new, there are a couple of things. One is DNA Gyrase. This DNA Gyrase, this one, or ARCA topoisomerase, the function is it moves in advance of helicase. This is helicase, and it moves in advance of helicase and relieves a strain and prevents supercoiling on the separated strands. So the function is removing the supercoiling structure. So once the helicase, if the helicase wants to separate these two strands, then the supercoiling structure should be relieved. Otherwise, the helicase cannot do at the same time relieving or uncoiling the supercoil and then separation. It does only separation and making the strand separated, not the removing the supercoiling structure. So one enzyme as Gyrase is doing the is removing the supercoil, and the other one, which is helicase, is separating. There is one more function that you can see over here. These are single-strand binding proteins. The function of single-strand binding proteins is somebody has to be there for not allowing that these strands after separation goes back to themselves again. So imagine that this helicase is moving forward, and then there is a possibility that then they are coming back together again. So it's like two things that they like each other, and then once you separate them, immediately they want to go back to each other. So someone should be standing over here and don't allow these two strands go back immediately, and that's the function of the single-strand binding proteins. They don't allow two strands go back together fast. I think I have already explained all the functions of the enzyme, including DNA ligase, which is functioning here by attaching or by ligation of the nucleotides at the place of the RNA primer removed, and then DNA polymerase 1, which is attaching the nucleotides over here after RNA primers removal. DNA polymerase 3, this is what we have already mentioned as the main DNA polymerase for adding polynucleotides, and DNA polymerase 1, and then RNA primase. So you can pause the video and read this page by yourself. I just move. It's the summary. I have already explained everything. And this page. So you can pause the video and study by yourself. It's just a summary. Yeah. I will end this video until here, and I will record the next topic in the new video. Thank you for watching.